So next, we're going to talk about the structure of DNA. DNA is made up out of many nucleotides. Every nucleotide consists of three things. It has a nitrogenous base, either a purine or a pyrimidine, and we'll talk more about those in a minute. It has a sugar, a pentose sugar. In the case of DNA, it is going to be deoxyribose, and that's where it gets its name, deoxyribonucleic acid. Another important part is a phosphate group. All DNA is going to have two nucleotides that have purine bases. These are going to be adenine, and we'll call this just with an A, guanine, which we'll mark with the G, and it has two nucleotides with pyrimidine bases, thymine with a T, and a cytosine, which we'll mark with a C. And I've indicated the structures here. On the left, you can see this is the generalized structure for a pyrimidine ring. Cytosine and thymine are both pyrimidines within DNA. Uracil we'll talk a little bit more about later, but uracil is going to be found in RNA. RNA does not have thymine, it has uracil instead. DNA does not have uracil. In terms of the purine ring, there's this double ring structure. Guanine and adenine are going to be examples of this. So let's look a little bit more closely at the differences in sugars between ribose and deoxyribose. The main difference is right here. If you look at this carbon, in ribose it's attached to a hydroxyl group or an OH group. The deoxyribose, in contrast, just has a hydrogen at this point. So just these minor differences can make really an important difference in terms of the genetic material. So if we put the three parts of these nucleotides together, you can see here it's got the phosphate group, here it's got the sugar, and here it has the nitrogenous base. You can look to see here how these are connected end over end to form the backbone of DNA. The backbone of DNA has resulting phosphate groups with a sugar group, a phosphate group, and a sugar group. And these go end over end to form the backbone of the structure. They're all going to go 5' to 3' when we're talking about things like DNA replication or transcription and things like that. What we're talking about here is this is going to be labeled as the 5' end, this is going to be labeled as the 3' end, and that will become more important later. You can see here that the nitrogenous bases have just been simplified. In this case, they're just marked as a T or a G, and you don't see their subsequent structure as purines or pyrimidines. So the actual amounts, the relative amounts of A's, T's, G's, and C's in the DNA actually vary from species to species, and most species do not have each nucleotide equal to 25% of the total. Chargaff came up with several rules, though, that allow us to be consistent among the different organisms. And what we find is that in each species, whatever the percentage of A is, it must equal T. And the reason for this is because A's are always going to bind to T's in the DNA double helix structure. Similarly, G's are always going to bind to C's. So whatever the percentage of G's is going to have to be the same as the C's. So because of this, the A's plus the G's together must equal 50% of the total, and the T's plus C's together must equal 50% of the total. So here's a sample problem.